Hi, today we completed a lab titled Investigation Tools and Measurements. If you are absent, this lab will be omitted. However, there are notes that we took in your notes notebook that you will need to make sure that you have completed. So when we look at the scientific method, we see that science is based on experiments. And there are different steps to the scientific method. And some people have 7 steps like I have written here, but you can combine steps to make less steps or even expand steps. It's not really so much the number of steps, is the process. So let's look at the first part of the scientific method, which is dealing with asking a question. Remember that questions are often based on observations, and observations can be qualitative or quantitative. When we have observations or data that are qualitative, those are going to be observations or data that is going to be a written description, like green leaves. I'm describing the color of the leaf. Whereas quantitative observations and data is going to be expressed as numbers. So there will be a numerical number. So for example, if I'm measuring the length of the table, it might be 70 centimeters. 70 is a number, so that would make it quantitative. Also, make sure that you understand what an observation is. Observations when you're using your 5 senses to perceive events. So your description of something you can touch, see, smell, taste, or hear. It also must be objective. In other words, it can't be an opinion. For example, I might make the observation that your car is wet because I can see droplets on it. Please don't confuse observations with inferences because a lot of students do. An inference is a guess or you're drawing a conclusion about something based on your observations. So again, when we look at our example, based on your car being wet, that first part is an observation. The inference that I'm making is I think you just washed your car. Now that's a logical inference, but washing your car, I don't know. I did not observe you washing your car. It might be that your car is wet because it rained. So again, if I ask for an observation, make sure you give me an observation and not an inference. Also, when we construct a hypothesis, remember a hypothesis is a testable prediction based on observations that describes a cause and effect relationship between variables, the variables being the independent variable and the dependent variable. A lot of hypotheses are written in an if-then format. Not all of them, but a lot of them. Now, when we carry out an experiment, we have to make sure that we identify the independent variable, which can be abbreviated IV. The independent variable is the thing the researcher will deliberately change or manipulate in the experiment. It is the only thing different between the experimental groups. So it's the one thing that's different between your groups. When you're graphing your data, the independent variable will go on the x-axis of the graph. The dependent variable, or DM, this is a thing that changes in response to the independent variable. It is usually represented by the data you collect in an investigation. In other words, it's whatever you're measuring. So when you're conducting your experiment, you're going to be measuring or observing something. Whatever that is your dependent variable. Again, when you're graphing, the dependent variable goes on the y-axis of the graph. The way to remember that about graphing is if you remember dry mix. The D in dry is representing the dependent variable, and it will go on the y-axis. The I in mix is representing the independent variable, which goes on the x-axis. Now, however many groups you have set up, the groups that receive the special treatment, meaning the independent variable, are going to be called your experimental groups. Your control group, which to me makes it a little bit confusing, can also be known as the experimental control. This is the group that does not get any special treatment. You will compare your experimental groups to the control group. So sometimes it's considered the normal group. It's going to serve as a baseline. Now, I have to admit, not all experiments have control groups. Now, everything about your groups should be the same except for that one independent variable. All the factors that are the same between the groups, all those things that are the same are going to be called constants or controlled variables. So the things between the experimental groups and the control group that you keep the same, those are called constants. You also should repeat your experiment. Repeated trials ensure the results aren't due to chance and ensure that the data is precise. Now, these scientific method notes are found on page 4. So if you will now turn to page 5, there is a doodle diagram where we're going to go over a specific example of trying to identify the independent and dependent variables, etc. So again, the first thing we do is observe and wonder. You observe nature and wonder about something. So for example, a scientist notices that carrot plants seem to grow differently in different soils. So our second thing is research. We're going to gather preliminary data and read about past research from other scientists. So a scientist reads about other experiments on carrot plants and learns that the nitrate concentration in the soil might matter. So that leads us to 3, the question. You're going to form a scientific question that is testable with an experiment. So for example, with the gathered information, a scientist asks this question: what is the optimal nitrate concentration for carrot growth? Optimal means best. Then you're going to form your hypothesis or educated guess to answer the scientific question. For example, a scientist hypothesizes that the more nitrate in the soil, the better the carrots will grow. And yes, you can put that in an if-then statement. If more nitrate is added to the soil, then there will be more growth. You'll then design and plan your experiment to test your hypothesis. Again, using past research to determine what temperature and what carrot seeds to use, the scientist designs an experiment to grow seeds in the same type of soil with varying amounts of nitrate added. You'll then conduct the experiment and repeat it for several trials. The scientist records how tall each carrot plant grows over the sequence of days. At the end, the scientist records how much each carrot weighs. Other observations, like how many flowers grow on each plant, is also recorded. The scientist will then analyze the data and decide if the data shows anything new or important. The scientist examines the data and calculates that the best amount of nitrate for carrot growth is around 10 parts per million. Too much or too less nitrate or too little nitrate slows down growth. So then we'll form a conclusion and then maybe even wonder and ask additional questions. Here you'll confirm, support, or reject your hypothesis and wonder about new ideas or experiments to further test scientific questions. So the scientist concludes that the data supports his hypothesis somewhat, but acknowledges that too much nitrate isn't good for growth. The scientist wonders what else in the soil could affect plant growth. The last thing is a doodle diagram on page 6, which is again still addressing this information. So again, the previous knowledge, we know that 5 parts per million of nitrate in the soil grows better than no nitrate added to the soil. When no nitrate is added, there's no growth. So we have our question: What is the optimal soil nitrate concentration for carrot growth? Our hypothesis, if more nitrate is added to the soil, then the carrots will grow more. So we have our setup, and let's pretend that we end up having 4 groups. Group 1, we added no nitrate to the soil. Group 2, we added 10 parts per million. Group 3, we added 100 parts per million. And group 4, 50 parts per million nitrate. Now, there is, and this is more of an AP biology topic, different types of controls, positive and negative. For our purposes, we're only going to really work with negative controls, and we'll just call it the control group. So again, a negative control, or what we're just going to call the control group, this is when the group doesn't have the independent variable or the special treatment, which in this case is nitrate. So again, no nitrate. So group 1, that's our control group. Group 2, 3, and 4 make up our experimental groups. So when we look at our experimental setup, our independent variable is, remember, the one thing that was different between the groups, which was the amount of nitrate in parts per million. The dependent variable is what we measured during the experiment. And what we measured during the experiment was the mass of the carrots in grams. Now, what did we keep the same between our 4 groups? Well, those would be called constants. We watered the groups the same. It was the same types of carrots, seeds. We use the same amount of soil and the same type of soil, except for the concentration of nitrate. And I even added a little sun. We obviously gave them the same amount of light. All of these things that we keep the same are constants.